Now we continue with second speaker, Prof. Bulent Kukurtcu, curriculum vitae, key subject, specialist biologist, scientific advisor, and technical scientific training, project management, and business development. Academic title from Turkey and Spain. And doctoral from Division Biology Department of Autonomous University of Madrid. And now Prof. Bulent will present the topic with immunomodulatory and antioxidant effect of nutraceutical with molecular activation technology. Time for presentation 20 minutes. Time is yours, Prof. Bulent.
So thank you very much. First of all, I would like to share the screen. Yes. Excuse me. First of all, thank you very much for the organization for inviting me, and thank you very much, Dr. Sri. And I would like to just add my point of view as a biologist and talking about the product that is exactly what Dr. Sri said, and just supporting her and with this product information. Before that, I would like to briefly talk about our company. It's a Spanish company established 40 years back. It's a company was born in 40 years back and producing different health products. We don't discover novel compounds, continuously patenting and developing this. So we have totally different model of business. What we do is we work with natural molecules, but very, very studied natural molecules in different pathologies. What we do is we have a technology, we call it molecular activation. We potentiate the therapeutical effects, biological indications, each of these molecules 10 times, 100 times, sometimes even more, depending the chemical structure of each of these compounds. Using our technology in our manufacturing process, in our development process, we apply good laboratory practices, good manufacturing practices, and the company has standard certifications like ISO 9001. The know-how, we call it molecular activation, it's a biocatalytic process and was born in CSIC. CSIC is one of the most important research center complex in Spain, from mathematics to engineering, from medicine to marine sciences. They have different research groups. Their biochemistry groups discovers this technology, and the same team with the same know-how became part of our company. And since then, we apply this technology to every single molecule. We can resume this technology with this picture. We have reactors. In these reactors, we produce an electrical field, but time and intensity depending to each of the chemical factors of these molecules. And as you know, every molecule has their own pH, own molecular weight, so on. And the process can be done in 2 hours, 3 hours, or 3 days, depending on these chemical factors. So the technology basically is inject or bond with electrons to the molecule in these reactors. And what it happens is, as you know, the molecules formed by atoms. Atom has nucleus and electrons around that. In orbits, in the internal orbits, when you inject electrons, there's an interchange or placement of these electrons. And during this replacement process, some they crash between them, and that produce energy, internal energy. And that energy potentiate activation, therapeutical property or biological indication of each of these molecules. So finally, you have the molecule with more energy, but with the same structure, because we don't destroy the molecule. We don't change the molecule. We are talking about the same number of electrons, same number of protons, the same structure, but with potentiate activity. Excuse me. We can give an example of this process with salicylic acid, for example. Salicylic acid is a secondary metabolite of Salix alba plant, and we use for ages this molecule for its properties like antipyretic, anti-inflammatory, revitalizant, but also it's a powerful antioxidant. Before the activation, we can detect this. And when we start with the process, in factor 1 and 5, we have the maximum antioxidant capacity of this molecule. After that one, if you go on with the process, we start to lose the property. Probably the molecule also can be changed. So that means that during the process, we control how we are sure about that this antioxidant capacity is increasing without changing the structure. And when the molecule is stable, we stop there and to have the maximum capacity of this molecule. What is antioxidant capacity? That is a process giving properties to the molecule. The molecule is more polar, has more capability of transport electrons, more active, more sensible. So we make more antioxidant particles in this reactor, because not all the particles has the same activity like the others. So in this case, we increase the number of active, more polar molecules. So that means that when you use any compound, any molecule, all the particles of this molecule has more activity. That's why antioxidant means that a molecule which donates an electron makes neutral or neutralize oxidant molecule, which is very reactive, very damage producer. So in the cells, when we have these antioxidant molecules with more energy, we are able to neutralize all of them and reduce the damages in the cells. So that is the mechanism basically. So this amplification could be in antioxidant molecules, antiviral molecules, immunomodulator molecules, depends on the type of the molecules that we use in our products. So thanks to this technology, instead of looking for new compounds, we increase their activities, and that gives us to use very low concentrations, because we amplify indication. Instead of using very high concentration in the long-term periods, we use very little, and that will give better results, and that give no side effects when we use our products. Because of that, we don't charge the organism with the excess of the molecules, so we don't produce any damaging effects on liver, on digestive system, and this and this. So no side effects at all, and we have a very fast action thanks to this technology, because the molecules has more capability to participate in metabolic activities. Also, we have, if you check generally our products, they are multi-molecular products. For example, Vucit product that we will talk today, it contains 12 active ingredients. So that molecules there, they are not there by chance or randomly. This is also the result of long-term studies in our company. All these molecules, after the molecular activation, one by one, they participate in the product, in the searching time, in the searching concentration for creating a chemical synergy. Thanks to this chemical synergy, they have even more activity, and that activity creates synergy also with the other therapeutical products that we use in our programs. So Virucid is a very known product, one of the most historical in our company. This product, the name Virucid is coming from virus de SIDA in Spanish sentence. That means AIDS virus. So one of the reasons that we created this product was initially against HIV. 30 years back, they didn't have good enough retrovirals, and the people were dying because of this disease. And as you know, like in other viruses, the virus doesn't kill directly. The virus creates secondary problems, and always this secondary problems create damages and make a difficult situation in the patients. In case of HIV, attack directly CD4 cells and defense immune cells. In case of COVID-19, you know perfectly, also attacks and creates important problems in the respiratory system. So on. The reason that we created this product is being able to reduce the viral load, but also to repair the damages that that virus was creating in different organs and different systems. So Dr. Luc Montagnier, a Nobel Prize, one of the co-discoverers of HIV virus, also collaborated with us and participated in creation of this product, Dr. Luc Montagnier. So finally, we created and we used in HIV, against HIV, but also in the following years, what we see that the product is not HIV depending product. It's active for all kind of virus, and we have studies, publications, and we use against papilloma, herpes. So not only for that, a little bit far from the viral environment. If there is any problem related with the immune system, also we use this product in psoriatic patients, in immunodepressive diseases, because it's powerful immunomodulator, but also hematopoietic, hepatoprotective, and powerful antioxidant. So I can show you different titles because of multimolecular environment formula of the product. So when we talk about the diseases, we have to talk also the oxidative stress. And we have many external factors, infectious diseases, chronic diseases, even the aging create an inflammatory response. Initially, leukocyte activation, oxidative stress generation, oxidative reactive species generation that activate in the first immune cells. But the constancy of these factors will create collateral damages if it's not under control. So in case of circulatory system problems and heart problems is very similar. We have lots of external factors, and against these external factors, we have oxidative stress, and oxidative stress will create inflammation. Inflammation means the muscle of endothelial of the heart will have a dysfunction if it's not regulated. As we can see, endothelial dysfunction can create other type of problems. We can go till thrombosis and finally multi problem in failure in different parts of the organ. So talking about the biosheet, what is it? It's for understanding, we have to check one by one on their molecules. What we see here, glycyrrhizinic acid, glucosamine, together with these two, they have antiviral properties, and malic acid generally is anti-infectious property. Altogether, we fight against viral infections. And these are powerful antioxidants. Together, malic acid, ascorbic acid, zinc, arginine, together, they are powerful antioxidants. Also, these three produce regenerate the blood cells. In all clinical trials and publications, we see that the product is highly hematopoietic. And also we have all these molecules, arginine, glucosamine, together with glycyrrhizinic acid and zinc. This chemical synergy gives us immunomodulation, not immunostimulation, immunomodulation. That means that we prepare the immune organs, immune system, in the moment that the organ or the organism receives stimuli, produce more enough immune cells. And this zinc together with calcium pantothenate is biocatalytic. But as you can see, repeating the name of glycyrrhizinic acid makes this molecule is the main ingredients of our product. here is the formula of the molecule and the active antiviral group. But beside this antiviral activity, the molecule has important functions like regenerative action or constructive repairing activity in different organs. In these studies, we can confirm how it's important glycyrrhizinic acid for the liver and kidney functioning when they have different failures or injuries, and also as antiviral. These studies confirm is glycyrrhizinic acid. We use hepatitis A, B, C, and also glycyrrhizin is important for influenza virus, SARS-related coronavirus, herpes family, flaviviruses, and respiratory syncytial virus. The mechanism, non-specific antiviral mechanism, works for all kind of virus, RNA or DNA type of virus. The first is related with the biological receptors. The molecule is able to bind directly to these biological receptors of the cells. In case of CD4 cells, CD4 receptors. In case of COVID-19, ACE2, that the scientists search till now, and we can find from the literature the mechanism of glycyrrhizin binding on these biological receptors, inhibiting the phosphorylation and blocking the penetration of virus in both routes. But another activity of the product is also blocking the division or multiplication of the infected cells by this multiplication of virus, by inhibiting nuclear factor kappa V of the infected cells. So we know from the studies that glycyrrhizin, like in the study, halts viral replication by inhibition of kinase P activity and irreversibly being a kinase P inhibitor. So that means that no direct relationship with the virus, is just creating a protection with healthy cells, means no resistance. Even after treatment, the activity will go on. That's why the scientists, when we started to have SARS-CoV-19, they suggested as a important target molecule that we can use by its multiple activity. As we can see here, it is binding directly to EC2 for preventing the SARS-CoV-19 by blocking the penetration. And also the molecule downregulates pro-inflammatory cytokines, inhibits accumulation of intracellular oxidative reactive species, inhibits thrombin, inhibits the hyperproduction of airways exudates, and also naturally produce endogenous interferon, generally as being as an anti-infectious activity. So there is one important activity of glycyrrhizin is in inhibition of high group mobility box 1 protein. This protein is very, very important protein. We know this that interacts with nucleosomes, transcription factor, and histones in the cells, and it has very important activity on bending the DNA and making a binding process with other proteins. But when it's presented extracellularly, it's as a mediator, activated macrophage monocyte secretes this amphoterine and as a cytokine mediator of inflammation. But after that, in the extracellular area, if antibodies cannot neutralize it, that confer protection, that creates damages and tissue injuries. It's not only in one disease, in every disease. For example, all related with metabolic disorders, obesity, hyperglycemia, we have important extracellular amphoterine level in organism. And also related with the respiratory system problem, even the allergies, we have these amphoterine here. And the scientists suggested at the beginning, in the first SARS, when we had SARS-1, they had this study already suggested this amphotericin as a target molecule against acute lung injuries. If we are able to neutralize this, and we are able to reduce the damages. This study shows that how glycyrrhizin directly binds to amphotericin for in secretional activity. And another study also confirms that the glycyrrhizin binds to amphotericin by two seats, and has an important activity on reducing inflammatory stimuli, and by that injuries. But it's related in myocarditis. Also, we have important activity or responsibility of amphotericin. Here, in case amphotericin expressed an experimental autoimmune myocarditis study, like this scientist suggested, and when they blockade the amphotericin, and cardiac fibrosis significantly elevated. And in this case, here the results that decreases also the collagen deposition and ameliorate fibrosis in this study. Another study also shows the effect of glycyrrhizin directly and the diseases, and the blockage of this molecule will be important example of the treatment of autoimmune myocarditis and inflammatory cardiomyopathy. And here also, this study suggests the protective effect of glycyrrhizin on myocardial ischemia, reperfusion, injury-induced oxidative stress. Here, in conclusion, the scientists found that glycyrrhizin attenuates myocardial IR injuries in rat, and protective effect of glycyrrhizin on oxidative stress and inflammatory reactions, which is they suggest as a novel therapeutic approach. Besides of this, of course, UCIT is not only glycyrrhizin. We have glucosamine. We have more activity, as Dr. Sri before explained, zinc and other molecules as important activities in this pathologies. But here also, glycyrrhizin showing an important protective effect against RNA type virus. By itself, BioSeed, we have many different studies in hepatology, in cardiology, in pediatric, in urology. I bring here some of them. For example, the use of BioSeed in the patients with non-alcoholic steatohepatitis patients, NASH, how is the product is able to prolong the life quality of the patients by reducing the problem in the fat accumulation. Here, the use of BioSeed as a powerful immunomodulator in psoriatic arthritis as a systemic product, together with other topic products against psoriatic arthritis. Here also, the publication in British Medical Journal, how the BioSeed is able to prolong the life expectancy of the patients with decompensated cirrhosis because of hepatitis C virus. In this trial also, they show that the product is able to regenerate the healthy tissues of the liver and make more functional liver. Another study also with hepatitis C people, how the product is a powerful adjuvant in the therapy with interferon and ribavirin by reducing the toxicity of ribavirin and assuring the therapy can go on with patients. That's why that is also they published in the journal. And another study also in fatty liver diseases, just in 6 months use of the product and exercises is able to reduce the fat accumulation in liver. But this study also shows, again, in hepatitis C people, how the product is important in the regulation of inflammation. In this study, the publication, the patients treated with VUSIT controlled TNF-alpha, which is pro-inflammatory cytokine, increased IL-10 anti-inflammatory interleukin, which regulates macrophage and dendritic cells activation, increased mean interferon gamma, interferon produced by NK and T cells, and that activate macrophage. And what we can see in the study that we have this regulation and normalization of all levels, ALT, AST levels also. So many studies, not only in human, but also in animals, shows the regenerative effect and normalization of the organ by this mechanism in liver, in heart, in kidney. We can share with you all these studies. One of the most important to know also related with the product is a powerful, right now is the most powerful antioxidant product in the worldwide market as a powerful antioxidant. This Brunswick laboratory is specialized on antioxidant products, molecules, showed in their analysis that the VUSIT has 12,000 ORAC value, means that oxygen capacity of absorbance of oxygen radical species just in 1 milliliter. That means that right now that makes the highest value ever registered in different products. That means that in any system, any organs related with oxidative stress, I'm able to enter in these cells and neutralize all these species and reduces the damages in the organs, in the kidney, in heart, in the digestive system related, all organs in kidney. So that is also important. We have studies, for example, using VUSIT in for improvement of the sperm quality in andrology, the men who cannot have babies because of the conditions, sperm production conditions. So in this case also, the VUSIT is a powerful antioxidant, helps to normalize this situation. Right now, VUSIT we use in the COVID-19 prevention and disease management and the recovery and the vaccination programs. This product has lots of studies also since 2020. For example, this one of these is VUSIT showed this publication recognized immunoregulator properties, including an important modulation of interferon gamma, TNF-alpha, interleukin-1 beta, and interleukin-6 levels in patients with infectious diseases. Doesn't matter, but in this case, important results with COVID-19. But we have this confirmation with other type of infectious diseases also. And that means that VUSIT is a model for SARS-CoV-19. Another study or publication also shows that efficiency of VUSIT in different symptoms that creates disease and the reduction of the hospitalization. So thank you very much. That was the general information about our product. I'm ready for the questions and answers. Thank you very much.
Thank you, Prof. Bulent. It's interesting presentation.
Thank you.